let's talk about how we can actually tell bacteria apart. We're going to pretend that we're the scientists or pathologists looking underneath the microscope to see these bacteria. So here's something that they might be looking at. And you can see that there are 5 examples. So let's just write down what we see. So vertically, in the top 2 at least, it looks purple. And on the right, I can see that they're pink. And there's this thing I can barely see down here, so I'm just not going to write a color for now. But vertically, purple and pink. So now let's look at the rows, and I can see that in the first row, they kind of look like little circles, kind of maybe spherical. So I'm just going to draw a circle here. And in the second row, I can see that they're more stretched out and kind of like a rod. So I'm just going to draw this kind of stretched out oval looking shape for a rod. And this bottom area I can barely see. So since I've been using a light microscope for the top view, I'm going to use the opposite. It's called dark field microscopy, but don't worry too much about the name. Just basically know that because these don't stain the same way, they have to be viewed differently. And the best thing I can call this is a squiggle. And I hope that makes sense. Let's go over that quickly one more time before we get into the official names for these things. So first row, I can see that they're round, and that's a sphere. And in the second row, it looks more like a stretched out rod looking shape. And in the third row, it kind of looks like a squiggle. So the single version of this is coccus, or a bunch of them are called a cocci, and a single rod is called a bacillus, whereas a bunch of them are called bacilli. And a single squiggle is called a spirochete, and the plural version is spirilla. So now we know about the 3 main shapes that bacteria can take, but we haven't talked too much about the color yet. And this color comes from a special stain called the Gram stain. And it basically stains the outside of the bacteria. And if it stains well, it stains purple, and if it gets washed off and stained with something else, it turns pink. And if it stains purple, we call that gram positive, whereas if it stains pink, the bacteria is called gram negative. So here I've done that with a positive sign and a negative sign. So we're going to take a closer look at this color aspect. So this might look familiar to you, but this is just the general bacterial structure that we've gone over before. And again, because the stain is only on the external layers of the bacteria, pay attention to just the external layer. So even though right here it says capsule, cell wall, and plasma membrane, you'll see in a minute that these layers are really different between gram-positive or gram-negative bacteria, which is shown here actually. So think of this as a zoomed in version of this peeled off bit. It does have the plasma membrane layer, a really thick cell wall layer, and a capsule layer. But for gram-negative bacteria, they actually have the plasma membrane again, but then they have a cell wall that is much thinner compared to gram-positive bacteria, and it's followed by another plasma membrane, and then a special layer called a lipopolysaccharide layer, and then a capsule. And don't worry that you didn't quite catch all that right now, because we're going to now draw them really zoomed in so you can see it. So here is that plasma membrane. And remember that it's just a lipid bilayer with some integral membrane proteins, which are drawn here in little blocks. And the unique bit is this really thick peptidoglycan layer. And I'm just going to write this out so you can see it. So peptidoglycan. So glycan just basically means sugar, like glucose. And these rods that you see here are made of these long chains of sugars. So that's the glycan part. And then there's peptido, which just means peptides, so your proteins. And those are just the connections between these rods that you see here. Oh, and I just realized this is a little bit of an optical illusion, but there you go. So it's almost like a brick wall with these kinds of connections. So that's where the word peptidoglycan layer comes from. It's just long sugar chains connected by proteins. And then the final layer is the capsule or the slime layer. So that's gram-positive bacteria. So now let's take a look at the other type of structure. You have this inner membrane, And then you have this very thin peptidoglycan layer. And again, this is made of sugars all along these rod areas. So I'm not going to write that out again because it's a super long word. It's going to take up space. Then you have an outer membrane. So here. And then next is this brown layer, also called the lipopolysaccharide layer, which most people abbreviate as LPS because lipopolysaccharide, LPS. It's also exactly what it sounds like. There is a lipo part, which is these lipid areas at the bottom, and polysaccharide, which are these sugar chains at the top, polysaccharides. And then this layer is covered by a capsule. And then there's a little bit of space next to the plasma membrane on both. So above this plasma membrane here and between these two plasma membranes over on this side. And this is just called the periplasmic space. So I'm just going to write it over here because it's easier. And again, think of it peri next to and plasmic, plasma membrane, so next to the plasma membrane space, between these two layers and above this layer before the peptidoglycan layer here. So now we can think about why this would show up purple and this one pink. So the Gram stain, when you first apply it to a bacteria, it just stains the whole thing purple. And then you'll wash it off. And because the Gram-negative bacteria has this very thin peptidoglycan layer in their cell wall, it washes right off. And later they'll restain it with something called safranin, which isn't important, but they come in and restain this to make sure that you can see it and it's pink. So that's Gram-negative. But because this Gram-positive bacteria has this super thick peptidoglycan layer, it will keep its purple stain and it will stay purple. So this should explain why these particular bacteria stain purple or pink. And now you should have a good understanding of one of the main ways to tell bacteria apart, both by their shape and color, and you should also understand where that color comes from because of their cell wall structure.